Good afternoon, students. Today I'm going to move forward. Today I'm going to move forward with the last part of our lecture of the protozoa. Today I'm going to discuss the ultrastructure and the economic importance of the protozoa. So before starting the ultrastructure, let me give you some of the idea and examples of the last classification of the protozoa which we discussed on Saturday. So okay, just going to revise and some of the basic information. Okay, as well as I have added some of the examples in each class. Okay, and as well as added the means of locomotion. So starting with the first phylum, Rhizopoda, and some of the examples of the Rhizopoda are amoeba, radiolaria, nagliria, and the means of locomotion is basically cytoplasmic streaming. Now moving forward, with the next one, Zoomastigophora. And the common examples are Trypanosoma, Leishmania, Giardia, Tricho, Nympha, and their means of locomotion is by flagella. Okay, and type of nutrition is heterotrophy. Some are parasitic, and some also absorb nutrients. Across the cell membranes. And the last phylum is a Sporozoa. Okay, is a Sporozoa. Common examples are Plasmodium and Toxoplasma. Their means of locomotion, okay, they basically live in the host. These sporozoans are, most of them are parasites. They are heterotrophs and again absorb nutrients across the cell membranes. So now moving towards the ultrastructure. Okay, this is just a diagram of a protozoan cell. Okay, and it is similar to that of eukaryotic cell. So basically this cell possesses most of the organelles of the eukaryotic system, but they differ from the plant cells. Okay, so protozoans, as we discussed in the last lecture, are basically single-celled eukaryotes. Okay, and the basic body organization, which is seen in the figure of the protozoan, basically consists of the external plasma membrane, which basically encloses cytoplasm and nucleus. And there may be one or more nuclei, and in some taxa, okay, nuclei are basically of two types: larger macro ones and smaller ones are micronuclei. Okay, some of the organelles which are present in the cytoplasm are visible through the light microscopy. And as we discussed in the last lecture, the vacuoles basically help in the uptake of the food. Okay, and basically the body which the protozoan contains, okay, or the cellular body, okay, which basically consists of basic ones: nucleus, cytoplasm, membrane. Okay, so this is a basic structure of the, but there is no presence of the chlorophyll. Now this one is a basic diagram of a paramecium. Now you can clearly see the cilia, okay, a micronucleus, a macronucleus. What are the functions of the micro and macronucleus? I will discuss now. Contractile vacuole, oral groove, food vacuole, anal pore, and this is a cytoplasm. This one is a cytoplasm. Okay, so different organelles are have basically different types of functions. Okay, so now moving forward with the lecture. Now the cytoplasm. Okay, now the cytoplasm uh, of the protozoa. In several forms of protozoa, pigments are diffused throughout the cytoplasm, and there may be numerous number of pigments. Okay, they can be green, brown, blue, purple, or rose-like color. Okay, and in the majority of the protozoa, cytoplasm is differentiated into ecto and endoplasm. Okay, ecto and endoplasm, and basically ectoplasm is more like of a gel, okay, and endoplasm is more voluminous and of fluids, and it made up of fluids. But the change from one layer, okay, to the another layer is basically a gradual one. Okay, and structures of the basically found in the endoplasm. Okay, are basically found in the endoplasm. Okay, and the cytoplasm, okay, as I told you, it may have number of colors depending on the pigments present. Okay, and it's an homogeneous, and the second term for the ectoplasm which is used is also known as cortex, and this cortex is made of this ectoplasm is made up of hyaline or gelatinous substances, and may be a part of cytoplasm bounded externally by the plasma membrane. Okay, so basically the ectoplasm may be defined as the part of the cytoplasm bounded externally by plasma membrane and internally by endoplasm. Okay, I am repeating the definition again. Ectoplasm may be defined as the part of the cytoplasm bounded and externally by the plasma membrane and internally by the endoplasm, and is made up of hyaline and gelatinous gel-like substances. In the case of Ciliophora, the cortex has a number of organelles. Okay, and as I told you, endoplasm is basically voluminous and filled with fluids. Okay, moving forward. Now the nucleus. Protozoan cells has at least one eukaryotic nucleus, and many protozoa however may have a multiple nuclei also. Okay, almost all ciliates throughout the greater part of the life cycle, and the protozoan nuclei are of various forms, sizes, and structures. And in several species, each organism has two similar nuclei. In the case of ciliates, two dissimilar nuclei are also present. One is the large macro and one is a small micronucleus one. Okay, so as I told you, now what is the function of macro and micronuclei? The function of the macronuclei is basically it controls metabolic activities. Okay, metabolic activities such as food, okay, various other growth factors needed, okay, whatever the enzymes are needed, okay, are the form and the structure and the size of the protozoan nucleus are basically extremely variable, and it's dependent purely on the type of protozoa, as most of the protozoa basically contain single some may have more than one nucleus also. Okay, and basically the macronucleus ones are also considered, as I told you, they are involved in the metabolic activities. They are involved in the metabolic activities. Okay, so they are also considered somatic or vegetative nucleus performing general metabolic activities. Okay, now macronucleus also offers variation in its form and structure. Okay, it is compact, it is generally compact, or even maybe spherical, or maybe ellipsoidal in most cases. But for example, in the case of Vorticella, it is elongated. Okay, in the case of Spirostrotum and Stantor, it is more like a chain. Okay, which is more like a chain type. Okay, which basically contains node-like structures. Okay, so the protozoan nucleus, okay, as a whole may be vesicular or compact, and the vesicular nucleus of a nuclear membrane, which is basically thin and delicate, but the nucleoplasm is very distinct and the chromatin content is less. Okay, now moving in the case of yes, I forgot to tell you, in the case of a amoeba like entamoeba, uh, the nucleus is basically composed of irregular masses and each containing a numerous small particles, and it disappears during the mitosis process. Okay, now comes the cysts. Okay, what are cysts? Okay, many protozoan form resistant structures. Okay, basically forms the resistant structures at certain times of their life cycle, especially when the conditions are not good for their growth. Especially when the conditions are not good for their growth. Okay, and the seeds are basically able to survive adverse environmental conditions such as desiccation, low nutrient supply, and even under the anaerobic conditions they help the protozoa to survive. Okay, as although the protozoa are basically aerobic ones. Okay, as all of the protozoa are basically aerobic ones. Okay, this is how the cyst help the protozoa to survive in the adverse conditions of desiccation, where there is low amount of water, low amount of nutrients, and even when the oxygen concentration in the environment is very low. The cyst forms. So there are various protozoa, okay, where disease is basically transmitted to a cyst forms. Okay, so basically this protozoa, there are certain diseases where a cyst form is basically transmitted from a one person to the another. For example, Giardia, Lambelia, Entamoeba. Okay, so there are more number of examples where a cyst is involved in the transfer of disease. And there are also other type of yeast. There are also other type of cysts which are basically reproductive ones, which are basically the reproductive ones. Okay, now comes the function of the cyst. Okay, I'm repeating the functions again. They protect against several unfavorable conditions, serve as a site of the multiplication, okay, attachment to the surfaces of the host in the case of parasitism, and the transmission stage of the protozoa is basically the cyst stage. Where they are transferred from one host to the another host. You see, as I give example, one of them is anta amoeba, and the second one is and the second one is giardia. Okay, now please have a look at the photograph of the cyst given here is of a giardia species, which is still undergoing a existment process. It is undergoing a existment process. Please have a look at the photograph given here. So basically this one is a flagellated vegetative cell called a trophocyte, and is basically emerging from the right of the cyst, okay, where you blue colored one, okay, blue colored one, okay, and the covering given here, okay, is of a giardia, okay, giardia, which causes disease in human beings. Now moving forward with the locomotory organelles. Okay, now moving forward with the locomotory organelles. Pseudopodia, okay, flagella, and cilia. In addition to this, few protozoa without such organelles can carry out gliding motility movement also. Okay, similar which we have studied in the case of bacterial gliding. So gliding, the mechanism is same, where a or one of the medium is basically used for the moving. Now what is pseudopodia? Okay, or a pseudopodia means singular ones, is basically a temporary projection, okay, is temporary projection of a part of a cytoplasm of those protozoa which do not have a rigid pellicle. Okay, I am repeating again, what is it? It's a temporary projection of a part of a cytoplasm, okay, whose of those protozoa which do not have a rigid pellicle. For example, this one is a random any of the protozoa. Okay, so what will they do? Now they will extend, okay, they will extend this cytoplasm, okay, they will extend or project the cell towards this direction, where similar to this type of structures are basically formed, okay, which helps them for the motility, okay, as well as to capture food, the food. Okay, so basically the sauropodia are characteristics of the amoebas, or the sarcodina. Okay, what are sarcodina? Basically rhizopods, okay, so they have two basic functions for the locomotion, and the secondary function is of food, okay, is of capturing food. Now some of the examples of different types of pseudopodia. There are also different types of pseudopodia. For example, lobopodia. Okay, for example, lobopodia. The diagram given here is of amoeba and arcella. Next one, again few examples of pseudopodia. Okay, in which three different types are given here: reticulopodia, philopodia, and lobopodia. Okay, this one is a globigerina, okay, and this one is a chlamydiophores, and this one is a difflugia, different types of pseudopodias, okay, reticulopodia, philopodia, and lobopodia. Now comes the flagella, okay, flagella or a flagellum singularly is an extremely fine filamentous extension of a cell. Okay, as a rule, the number of flagella present in an individual protozoan varies from 1 to 8. Where 1 or 2 is the most frequent number found in the protozoa, and this flagellum is basically composed of two parts. An elastic filament called axoneme, and a contractile cytoplasmic sheet that surrounds a axoneme. Okay, so basically two parts, axoneme and a contractile cytoplasmic sheet, okay, which basically surrounds the axoneme. Okay, for example, let us assume that's an protozoa, okay, This one is a protozoa, and basically exonym is this one is a elastic filament. Okay, this one is a elastic filament. Okay, now a covering, okay, around is basically a covering around is basically of a contractile cytoplasmic sheath. Okay, now moving forward with the cilia.